This is the fifth day in our genomics introductory course, and here we'll be taking us further on the introduction to databases. And we want to know the gene prediction tools, the types of database. We've explained that, and data quality and data uses. We will just take us through the gene prediction tools, which is important to what we want to achieve. Now, the gene prediction tools, although we'll be taking us through that in hands-on session, it refers to the process of identifying the regions of genomic DNA that encodes genes. This includes protein coding genes as well as RNA genes. So gene finding is one of the first and most important steps in understanding the genome of a species once it has been sequenced. And gene prediction is one of the key steps in genome annotation. When you annotate a gene, it is one of the process of gene prediction. So following sequence assembly, the filtering of non-coding regions, and repeat masking. So it is closely related to target search problem. And many aspects of structural gene predictions are based on correct understanding of underlying biochemical processes in the cells. So to explain further, as you can see here, now we know what an open reading frame is, and you can see that one of the key components of gene annotation is knowing where the right reading frame is. We have a set of sequences, as you can see in this figure, A, G, T, A, C, G, T, T, T. But we don't know where the gene is. You can start reading the gene from where we can find the gene, where we can predict it from. So we started from the first one. We saw, oh, this is the wrong prediction. Second, from G, wrong prediction. It was when we started from T that we found the start codon of that particular gene, and we reached the stop codon of that particular gene. So that means open reading frame 3 is the right reading frame to start reading your gene of interest from. Then gene annotation is the process of identifying the locations of genes and all of the coding regions in the genome and determining what those genes do. It is important. We are not just looking at the location, we are looking at what that gene does. And we've said earlier that it is based on our pre-existing understanding of what that particular gene does, as in the case of insulin and other gene of interest. Then the gene annotation consists of 3 main steps. The first one is identifying the location that does not code for proteins. The second part is identifying elements of the gene, which is the gene prediction that we explained earlier. And then the third part is attaching biological information to that element. So there are different methods that you can use. One of such is BLAST, Basic Local Alignment Search Tool. The other part, you can use genome browsers like Ensemble to look for the gene, to annotate for that particular gene. And then structural annotation consists of genomic elements such as ORFs and the likes. And then there are different gene annotation tools. I've listed them here. We have encode, entries, Ensemble, gene code, gene annotaology consortium, gene RIF, RefSeq, Uniprot, and Vega. There are lots of gene annotation tools, but these are some of the examples.